Good morning students, welcome back. So today we are going to clear 2 to 3 practicals, and after that we are going to complete the whole experiment of this semester. So we have already discussed few practicals related to this semester. Only 2 to 3 experiments are left. So we are going to discuss about the experiment throughout this semester. So what are the different experiments? You know that each and every paper is consisting of minor experiments as well as the major experiment. So this is from paper 1, ecology and ecosystem. We can find out one will be the minor experiment, other will be the major experiment. In the regular classes, or when you are taking the MSc degree regularly, then that times the practical was consisting of 50 marks. So today also you will be allotted 50 marks for the practical. So here the minor experiment, regular paper wise, the minor experiment they used to carry the 15 marks, and major experiment it used to carry the 25 marks. Altogether this is 40 marks. 5 marks are allotted for the journal and 5 marks for the viva. So altogether you can say that the 50 marks is allotted for the practical. So here we are going to conduct the experiment by means of the Google form. So by means of Google form we are going to conduct the experiment, and we are going to discuss today what are the different topic which is left, or which are the different experiment till now we have not discussed. So normally this Google form is consisting of the MCQs questions. So you have already attempted the theory papers. Similarly you have to attempt this Google form MCQs questions for the practical, and it is consisting of the 40 marks. Later on it is converted into the 50 marks. So MCQs altogether, each paper 20 MCQs will be given. We have to attempt all these 20 MCQs and submit the form, and then it is later on assessed. So coming up to the minor experiment, the first one, that is determination of diversity indices in plant community. This is the experiment which is left. Next is that we are not going to perform this experiment. Next is determination of chlorophyll content from the plant species. I hope this experiment is already discussed. Determination of harvest method from the plant species. This experiment is left. I hope we have already discussed about the determination of harvest method from the plant species, but still if you want I am going to repeat it once again. Next, coming up to the major experiment. Here the major experiment, it consisting of the first one, that is IVI, means important value index, first one. Next, to compare two plant communities. I think I have already sent you the yesterday's practical. Compare how to compare the two plant communities. Next is the we are not going to perform again this experiment, that is this experiment. So we are going to perform this and this, or the MCQs will be based on this experiment. Next, coming up to the paper 2. So the paper 2, again it is consisting of the minor experiment as well as the major experiment. The minor experiment we have already completed this biogeochemical cycle or biogeographical zones of India, and the expense of water territory, मतलब water territory कहाँ throughout India में कहाँ पे spread है. So at least if you have not covered the expense of water territory, but we have already completed the biogeographical zones of the India. Next is identification and description of plant species. So if any plant is given to you, you have to take the LS of the flower. कोई भी plant का हमें LS लेना पड़ेगा, LS of the flower, and then we have to write down how many petals, sepals, stamen, stigma, etc. are there, और ovary का जो पूरा डिस्कशन है वो लिखना पड़ता है. इफ इट इज hypothalamus or the epigynos etc etc. So that was related to identify. We are not going to perform this experiment. So we have already done this experiment. We have done this experiment, that is plot the biosphere reserves on map of India. So on the map of India we are not going to plot it, but MCQs related to the biosphere reserves will be given to you. Next is prepare document endemic and exotic species of the plant and animal species for a selected pan. So in a selected pan, you have to select a particular protected area network. Consider if you have selected the Kaziranga protected area network. So in that Kaziranga, how many type of endemic species are there? और कुछ ऐसे species होते हैं जो कि उस जगह की native होते हैं, and certain species which are एक्सोटिक स्पीशीज, मतलब ऐसे स्पीशीज जो कि बाहर से लाए गए और वहाँ पे प्लेस किया गया है, देन दे आर सेड टू बी दी एक्सोटिक स्पीशीज. तो कोई भी एक सेलेक्टेड पैन में एंडेमिक स्पीशीज, वहाँ की नेटिव स्पीशीज कौन कौन से हैं और बाहर के स्पीशीज कौन कौन से हैं, वो फाइंड आउट करना पड़ता है. नेक्स्ट इज दी मेजर एक्सपेरिमेंट. इन दैट मेजर एक्सपेरिमेंट, first is the indicate distribution range of plant and animal species identifies as endangered on Indian map. So India map, on the map of India, what are the different type of endangered species of plants as well as animals. So we have already discussed this point. Next is the prepare a map of Maharashtra showing protected area network. So what are the different type of protected area network, that is national park थ्रू आउट महाराष्ट्र कितने हैं, या फिर वाइल्ड लाइफ सेंचुरी थ्रू आउट महाराष्ट्र कितने हैं, या फिर प्रोटेक्टेड एरिया नेटवर्क्स ज़रा होंगे तो कितने हैं. तो नेशनल पार्क, वाइल्ड लाइफ सेंचुरी या फिर प्रोजेक्ट एरियाज एक्सेट्रा, हाउ मेनी आर देयर, दैट वी हैव टू शो ऑन द मैप. सो सर्टेन MCQs विल बी बेस्ड ऑन द महाराष्ट्र प्रोटेक्टेड एरिया नेटवर्क. नेक्स्ट इज द this third experiment, which we are not going to perform it or discuss it, or this topic will be avoided. Next experiment is to study the plant community by using the line transect, using line, belt and profile transect. So here we have to find out the line, belt and profile, but still we are not going to perform this experiment. So all together we are going to discuss this portion, that is paper 1 minor experiment, major experiment, and the paper 2, that is minor experiment as well as the major experiment. Paper 3, that is determination of total organic matter in the soil. Here whole description related to soil is given. So a soil must have a particular amount of organic matter. So how to estimate the total organic matter in the soil? Next is the pH of different type of soil. So how to find out the pH of soil? So if a particular soil is given to you, so how you are going to perform the experiment, or how you are going to find out the pH of the soil, which we are going to discuss today. Next is the water holding capacity. How to find out the water holding capacity of a soil. And the hydrological cycle, that is water cycle. Next is the determination of mechanical composition of soil by pipette method. So if a particular soil is given to you, what is the mechanical composition, मतलब कि उसमें sand कितना है, उसमें silt कितना है, etc. can be calculated by means of the pipette, international pipette method. Next is the determination of sodium absorption ratio value of the soil. You know that the soil used to absorb certain amount of cation as well as the anion ions, and this work or function is done by means of the xylem. So xylem, what it does, it absorbs the minerals, either maybe the cations or anions, with the water, and then it is provided to rest part of the plant. So how this conduction is done, if a particular soil has high amount of cation or anion exchange system, then this absorption capacity can be, you can say that, influenced, or it can be increased. So sodium absorption ratio, amount of sodium, what is the amount of sodium can be absorbed by a means of soil, that can be discussed in this experiment. And the next paper, that is say the fourth paper, here we have to find out the total dissolved solids. Total dissolved solids means a water, consider a water, if water may be the drinking water resources, consider. So if this water is present, this water may contain certain dissolved matter and certain undissolved matter. उस अनडिजॉल्व मैटर को हम सस्पेंडेड मैटर बोलते हैं। तो कुछ मैटर ऐसे होंगे जो कि सस्पेंडेड होंगे और कुछ ऐसे मैटर होंगे कि जो डिजॉल्व होंगे। तो डिजॉल्व मैटर कैन नॉट बी सीन बट सस्पेंडेड मैटर कैन बी सीन। सो दिस सस्पेंडेड पार्टिकल्स जो भी प्रेजेंट है इस वाटर में, तो उस वाटर को हम क्या करेंगे, फिल्टर कर लेंगे by means of funnel and filter paper. So this is a funnel in which the filter paper is kept. Now we are going to pour the water. So when we used to pour the water, the suspended particles जो भी होंगे, they used to stick on the filter paper. इस filter paper पर stick हो जाएंगे, and rest the dissolved matter, water containing the dissolved matter, it seeps into the beaker. beaker में आ जाते हैं। तो now we have to take this amount, that is 50. First we have to take the 50 ml of the water sample, then we have to filter it, and after filtering we have to take this 50 ml in a china dish और petri dish बोलते हैं हम उसे। So in this china dish we have to add this filtered 50 ml of the water. But before adding, we have to first wipe it out, clean it, and then we have to first weight it. So we have to first take out the weight of the china dish, or we used to say that dishes, china dishes. So we have to first weight it. The first weight will be considered if it is 56.5 grams. And later we have to add 50 ml of this filtrate, and then we have to heat it. हीट करने के बाद ऑन ए हॉट वाटर प्लेट वी हैव टू हीट इट। हीट करने के बाद एक ऐसा कंडीशन आएगा, एक ऐसा स्टेज आएगा जहाँ पे पूरा पानी ड्राई हो जाता है, या फिर इट्स एंड पॉइंट यू स्विच ऑफ द होल वाटर और गेट्स इवेपोरेटेड। तो उस पॉइंट पे हमें क्या करना पड़ेगा, हॉट प्लेट को बंद करना पड़ेगा, एंड आफ्टर इवेपोरेशन सर्टेन अमाउंट ऑफ सॉलिड मैटर, डिजॉल्व मैटर जो होगा, दे यूज टू स्टिक टू द सरफेस ऑफ द पेट्री डिश, यहाँ पे वो, और इट विल ऐड द वेट, और इसके कारण क्या हो जाए, वेट बढ़ जाएगा। सो अर्लियर इफ द वेट वाज 56.5, लेटर इट विल बी 56.8, and this weight is very minimum in case of the river water or the drinking water resources. So you have to perform this experiment very carefully. Then what we are going to do is that we are going to subtract this, that is the amount of the dissolved matter which is present in the petri dish is that is minus 56.8 minus 56.5. So 0.3 will be the weight, and according to that weight we have to apply it on the formula, and then you will come to know the dissolved matter in the given water sample. Next is determine the hardness of a well water. How to find out the hardness, we have already discussed it. Next is the density flux by lux meter, and next is the noise pollution. Lux meter I hope we have already discussed it. Noise pollution is rest, which we are going to discuss. Next, coming up to the major experiment. This major experiment is consisting of determination of carbon dioxide, certain MCQs related to the carbon dioxide, what is the percentage of carbon dioxide in the atmosphere, what is the role of carbon dioxide. Next is the physical determination of physical parameter of soil or water. So in this experiment we are going to perform the experiment related to the water. You know that the well water quality is totally different from industrial wastewater. Again, the river water is different from the sea water. Here you can find out, in case of sea water, the dissolved matter is highest, because here the salt they used to normally dissolve in the water. So sea water, here you can find out the parameters totally they used to differ. But river water and well water is almost the parameter, almost they are or in almost in the range. और इफ़ यू कैन फाइंड आउट द इंडस्ट्रियल वेस्ट वाटर इज टोटली डिफरेंट. सो इधर पे हमें क्या करना है, तो यहाँ पे हमें हर एक डिफरेंट सैम्पल्स हमें दिए हुए हैं. वी जस्ट हैव टू फाइंड आउट कि हाँ टोटल डिसॉल्व सॉलिड्स सी वाटर में कितना है, रिवर वाटर में कितना है, इंडस्ट्रियल में कितना है और वेल वाटर में कितना है. pH ऑफ द वेल वाटर कितना है, pH ऑफ रिवर कितना है, pH ऑफ द सी वाटर क्या है. नेक्स्ट, व्हाट आर द फिजिकल पैरामीटर्स. कौन कौन से फिजिकल पैरामीटर्स हो जाते हैं, तो pH हो गया, फिर टेम्परेचर हो गया, फिर डिजॉल्व मैटर हो गया, डिजॉल्व टोटल डिजॉल्व सॉलिड्स हो गया, डिजॉल्व सॉलिड्स हो गया, या फिर हम सस्पेंडेड सॉलिड्स बोलते हैं, सस्पेंडेड सॉलिड्स हो गया. ये सारे फिजिकल मैटर हैं जो कि वाटर में प्रेजेंट रहते हैं, विच वी हैव टू एस्टिमेट इट. नेक्स्ट इज द डिटर्मिनेशन ऑफ डिसॉल्व ऑक्सीजन फ्रॉम द सी वाटर बाय Brinkland मेथड. How to find out the dissolved oxygen and how to find out the chemical oxygen demand. So we have already discussed the chemical oxygen demand. What is left is the dissolved oxygen, which we are going to discuss it today, and by the evening we are going to send I am going to send you the experiment videos, which all experiments are left. So till now we have discussed all this experiment, and what is left, which I am going to send you today evening. So thank you for watching. See you tomorrow with the next video.